Very excited, Nicole, about your talk, your presence.
Thank you. It's very nice to see you.
Is everyone, Jen, is everyone in from the waiting room? Should I start?
Yep, you're all set.
No? Okay, so I'll go ahead and introduce Nicole and we can get started. So I'm very excited to have Nicole King here today for the seminar, which was postponed from September. So I'm even more excited that we were able to reschedule it. So Nicole is an HHMI investigator and a professor of molecular and cell biology at UC Berkeley. She's before I get in, she'll tell you about some of the current things that she's working on. She did her PhD actually in gene regulation in Drosophila, I believe. And
that was my undergrad.
Your undergrad, okay.
That was my honors thesis, yeah.
I was trying to interpret the CV.
Yeah.
Okay, so she started in Drosophila and then did a postdoc with Rich Losick, where you had at least one publication
PhD.
Okay, I'm on. So okay. Anyway, you have at least one publication on Bacillus subtilis there.
That's right. Yeah.
Then a postdoc with Sean Carroll. Am I right on that? Okay.
Yeah.
There got into sort of founding the field of choanoflagellate biology and using that to dissect the origins of multicellularity in animals. And so this has been just an amazing series of studies that, is something to kind of admire and live up to, that has been both technically and conceptually innovative in setting up Monocyga brevicollis as a model organism and sort of establishing what this can tell us about how animals became multicellular originally. And one of the sort of amazing things about, Nicole's body of work is this ability to balance sort of small biology and big biology and think both about the molecular and genomic insights, as well as the sort of evolutionary and organismal insights that you can get out of this system, which I find really inspiring and has just been an amazing set of publications to read. And has given us a glimpse into how animals became animals, which is also just really cool. She's, of course, accumulated many honors, including Pew Scholar. She was a MacArthur Fellow. She's a member of the French Academy of Sciences, as well as the US National Academy of Sciences. And in addition to all of the scientific innovation, well, this is scientific as well, but I think has been an example of how to set up sort of a new field and make it ensure that it's open and sort of supportive scientific culture, which is also very inspiring. So with that, I'll turn it over to her, and I'm looking forward to a really exciting lecture.
Thank you so much. It's a real pleasure to be talking to all of you. I wish I was there in person. Maybe I'll just riff off of a couple of things that Luma just said. One is actually when I started studying choanoflagellates, Peter Holland and I organized a small meeting. I think there were only 11 of us, and we all agreed that we would be better. The field would grow and thrive if we all worked together and supported each other and collaborated. And so right from the bat, that was part of the culture. And we've continued to meet every 2 years, and the community has only grown, and it's just a wonderful, friendly, supportive group. So that's really been great. The second thing I would say is really for the trainees, and that is that this talk today is going to be about our single-minded pursuit of one question. And what I'm going to show you is that single mind, we have continued to fail in our single-minded pursuit of that question. And yet along the way, I think have made some really exciting discoveries. So, and that's sort of something I learned from Rich Losick. And that is that if you keep your eyes open and your mind open and don't ignore the unexpected results, that you can be taken in really exciting new directions. And so please don't judge me at the end of this talk when you see that we actually haven't accomplished our original goal, but instead hopefully glory with us in the other findings that we've made along the way. So, so today, actually, I've changed the title, and I'm going to be focusing on the phenomenon of mating in choanoflagellates. And this is interesting because one of the first incidents of cell differentiation that's thought to have evolved in the animal cell lineage or animal lineage was the differentiation of somatic cells from gametes. And so by studying the phenomenon of mating in unicellular organisms, we hope to gain some insight into how that process originally evolved on the animal stem lineage. Okay, so let me just back out and give you some big picture. I think many of us are fascinated by the incredible diversity of animals and animal forms. And animals really are, you know, I think of them as variations on this theme of multicellularity. And it's incredible, you know, we have animals swimming in the oceans and flying and, you know, crawling across the earth. And they make a living in very different ways, you know, some of them are filter feeders, some of them are predators. The diversity is remarkable, and it really has been a major focus of the field of evolution and development. How do we get so much diversity? But there's something interesting about animals, and that is that they all share a single common ancestor. And in a way, we see that reflected in the way that animals develop. They all start life as a single cell, the zygote. And through serial rounds of cell division, with the cells remaining clonal, largely having the same genomes, they arrive at these very different forms. And so really, animal life, every generation is a transition to multicellularity. Now, that's at the level of the individual, but in my lab, we're interested in the big picture. How did animals evolve multicellularity in the first place? Can we reconstruct the biology of the last common ancestor of animals and then back up in time and gain some insight into how multicellularity itself first evolved on the lineage leading to animals, and how mechanisms for regulating cell differentiation and the coordination of function within the multicellular animal, how did that evolve? And I won't go into details, but let me just say that this transition to multicellularity has happened over and over again within eukaryotes. So for the purposes of this talk, we're focusing specifically on the transition to multicellularity that led to animals. Now, had it been possible, I would have loved to have done this work by referring to the fossil record, because there you have an absolute, you're actually looking back at time. But unfortunately, the fossil record cannot or does not preserve the small, soft-bodied organisms that were the progenitors of animals. And even if it did, we wouldn't necessarily know it. And even if we knew it, it wouldn't give us insight into cellular and molecular mechanism. So instead, what we've done is turn to living organisms as a window into the past. And in particular, we're focusing on a group of organisms called the choanoflagellates. And the reason is that these are the closest living relatives of animals. And recently, by sequencing the genomes and transcriptomes of many different choanoflagellates, we've learned that they encompass a remarkably diverse group whose genetic diversity actually rivals that of animals. And so I've, you know, intentionally drawn these boxes to be approximately the same size. And so just as we can compare the biology of animals and gain insight into their last common ancestor, we can compare the biology of choanoflagellates to each other and learn something about their last common ancestor. And then pulling these comparisons back in time, we can learn something about the organism from which they evolved and think about what might have changed along the stem lineage that separates animals from all other life. Pardon me. So if you haven't looked at a choanoflagellate before, let me just give you a quick introduction. The cell body of a choanoflagellate is about 5 microns, so it's about the size of a yeast cell. And every choanoflagellate has an actin-filled collar of microvilli that extends from the apical pole and a long flagellum. And you can better see this in a live image. This is a high-speed camera or a high-speed imaging that we've slowed down. And what happens is that flagellum beats back and forth inside of the collar, and that generates currents, either propelling the choanoflagellate through the water column to swim, or to pull material out of the water column up against the collar, which allows it to capture it. And so they actually are really voracious bacteriivores. They're an important part of the food web in the oceans for essentially capturing bacterial prey and making that carbon available to larger sized organisms. So I mentioned that choanoflagellates are incredibly diverse, but there were very few people studying them. So we've really focused on developing a small number of model choanoflagellates that are experimentally tractable. And the star of this talk is going to be the choanoflagellate S. rosetta, which we named for the fact that it makes these really beautiful rosette-shaped colonies. And these colonies are, to me, reminiscent of early stages of animal embryogenesis. So we were very interested in understanding how these form at the same level of detail as we see when we study animal embryogenesis, okay? And so over the decades, starting, first with the description of choanoflagellates, but then really taking off with the sequencing of the first choanoflagellate genome that was published in 2008, we've been working to develop tools to make S. rosetta and other choanoflagellates experimentally tractable. And so, for instance, with S. rosetta, we can control cell state transitions in the laboratory. We've established forward genetics, so we can isolate mutants. In 2018, David Booth developed a technique for transfecting choanoflagellates, which was remarkably difficult to get off the ground. And then in 2019, Pavel Burkhardt's group published a 3D ultrastructure of S. rosetta, including colonies. And so this has helped to reveal, in particular, the secretory pathway and how that works, polarized secretion in choanoflagellates. And then very excitingly, in 2020, so very recently, David Booth, while still in my lab, established CRISPR genome editing, and that has been foundational. And it's remarkable, again, so hard. We were working on it since I started my lab, getting some sort of genome editing going. And now with its establishment, it's just incredible, all the things that are being done, not just in my lab, but in other choanoflagellate labs as well. So I think it's really going to accelerate research on these organisms in the years to come. So why, I told you choanoflagellates are closely related to animals, but why should we be particularly interested in them? And one of the reasons is that they help us gain insight into the ancestral functions of genes that are critical for animal biology. Now, when I started the lab, I was particularly interested in identifying the animal innovations. What is it that distinguishes animals from everything else? And these are the types of genes you see. These are many, famous developmental regulators, for instance. But what was surprising is that many of the genes that were in textbooks as being animal genes are actually conserved in choanoflagellates. And so that means that they were also present in the last common ancestor with choanoflagellates. And these include things like C-type lectins, adhesion proteins like cadherins and integrins. There are collagen repeats. And I'm particularly, later in the talk, going to focus on G protein coupled receptors. But this is just to make the point that some of your favorite genes may actually be in choanoflagellates. And that raises interesting questions about what they might be doing at a unicellular level before they became involved in obligate multicellularity in animals. Okay, so what I'm going to do is first give you a very brief introduction to the life history of S. rosetta to give you some context for the biology that I'll be discussing. I'm then going to describe two unexpected discoveries that we made in past years. And these are that bacteria regulate both mating and multicellularity in choanoflagellates. And I'm essentially, even though that work has been published, I'm essentially introducing it again here because it motivates our study of choanoflagellate G protein coupled receptors. And this is very exciting new work, really more data are coming in every day. And so, this morning I was adding in new slides. So I'm excited to tell you about that. Okay. So as I mentioned, S. rosetta can form colonies, but it also makes a very happy living as a single-celled organism. And so one of the interesting things about it is how does it transition from being single-celled to colonial? And it does this through a process of serial cell division, just like a zygote dividing over and over again to become an early stage embryo. But these are highly polarized cells. And so what happens is they divide along the apical basal, not talking well, apical basal axis. And then the two cells will remain together. And this will continue with the cells reorienting to establish a two-dimensional, essentially, plate. And then they will pop out into the third dimension with further rounds of cell division. And you can see that here, this is a founding cell. And then through multiple rounds of division, it produces this three-dimensional rosette. So we've been very interested in understanding this. And one thing you might be wondering is how do these cells stay connected? And one way they stay connected is that through incomplete cytokinesis. They leave behind an intercellular bridge. And another thing that happens is here's a TEM of one of these intercellular bridges showing these mysterious plates that we've yet to identify. But one of the other things they do is they secrete extracellular matrix into the middle. And I'll show you in a moment that genetic screens have shown that key members of the extracellular matrix are essential for rosette development. So we think it's these two features, these bridges and the extracellular matrix, that are essential for building the rosette. But for now, I want to really focus on this question of regulation. So what is it that controls whether these cells will continue to divide as single cells or whether they'll switch into a pathway that leads to multicellular development? And of course, we're interested in both extrinsic cues and also the genetics that contributes to this transition. But when I first started studying S. rosetta, I was really frustrated in this effort. And the reason is that although it was isolated from nature as a rosette, when I brought it back into the lab, it was primarily in the single-celled form. So hopefully you can see these single cells. And you can probably also see the bacterial prey that we have to grow them with. There has to be live bacteria there for them to grow. And so this was exceedingly frustrating. Actually, my postdoctoral fellowship the proposal was that I was going to separate colonies from single cells and somehow, for the older members of the audience, I was going to use subtractive hybridization to identify genes that were specifically upregulated in one cell type and not the other. Younger ones don't even know what that is anymore. Anyway, I could never get enough colonial material to do anything. And so really kind of set it on the back burner until I came to Berkeley. And then at Berkeley, we started working on trying to get control over the bacterial diversity for genome sequencing. And interestingly, one cocktail of antibiotics we used actually preserved enough bacteria for them to eat, but blocked all rosette development. So in this condition, we never saw rosettes. And that raised the interesting possibility that cues from the environmental bacteria may help to induce rosette development. And that actually turned out to be the case. So that was, we had been changing all these other features of the culturing conditions, pH, various nutrients that we were adding in, temperature, you name it, we were trying it. But despite having trained in a bacterial genetics lab, it didn't occur to me that bacteria might be the key feature. And so this result got us thinking about the bacteria. And so we isolated different species from the environmental condition and added them one at a time here. And it turned out that one of the cultures was sufficient, one of the bacteria was sufficient to rescue colony development. And this changed everything. First of all, it emphasized the importance of host microbe interactions in the life history of S. rosetta. And secondly, it gave us really straightforward control over this process in the laboratory. So the responsible party here is this bacterium, Algorophagus, which makes these really beautiful orange colonies on plates. And this was exciting because it was co-isolated with the choanoflagellate. So, so it seemed to be an ecologically relevant partner. We could grow cultures just with S. rosetta and Algorophagus as prey, and it was sufficient food. And it's related to bacteria in the vertebrate gut. And so what we learned about the interaction here might actually have some relevance for interactions between vertebrates and their gut microbiota. So we definitely were very excited about this. And we collaborated with a chemical ecology lab, John Clardy at Harvard Medical School, to identify the compound that regulates colony development. And this turned out to be an extremely difficult process, largely because, well, for two reasons. One is that the inducing compound is a lipid, and it was really tricky to separate it. And the second is that it's present at exceedingly small levels in the bacteria. And so we had to grow up, we, meaning the Clardy lab, had to grow up vast quantities of Algorophagus to get enough material. But it was worth it because this is a fascinating molecule. This is what we've named RIF1 for rosette inducing factor 1. And it's a member of a class of molecules called sulfonolipids, named for the sulfonic acid head group. But these are interestingly related to sphingolipids, which are a critical signaling compound in animals. And again, somewhat poorly understood in terms of how this signaling works. So we definitely wanted to pursue this further. And so the question that has really been driving us, and the one that I mentioned that we have still not succeeded in answering, is what is the receptor for the rosette-inducing factor? How do choanoflagellates sense these rosette-inducing factors, and how does that signal get transduced to switch from being unicellular to colonial? And so what I'm going to tell you is our persistent effort to identify the receptors for the rosette-inducing factors, and then how that's led us in a number of really exciting directions, none of which have actually answered this question. So there's the setup. Okay, so the first approach was spearheaded by a really brave graduate student, Tara Levin, who came to the lab and said she wanted to set up forward genetics in choanoflagellates. And I thought that was a spectacular idea. You know, I think of forward genetic screens as the gold standard for linking genes to phenotypes. But there were some really critical barriers that I thought were going to prevent her from success. And I told her, I said, you know, as your mentor, I'm not sure that I can, in good conscience, encourage you to do this because I don't think it's going to work. And she broke it down. She's like, these are the barriers and this is what I'm going to do to overcome them. And sure enough, she succeeded on every one of those. So it was a real tour de force. So this is the way the screen worked. She would grow up, you know, one key challenge is that the choanoflagellates don't grow on solid plates. So they have to be grown in liquid media. And what that means is that it's not straightforward to isolate clones. But what she would do is mutagenize in liquid media, then quickly isolate clones by limiting dilution into 96 well plates. And these 96 well plates contained the rosette inducing factor. And so then she could visually screen through each of the plates, find the wells that had choanoflagellates in them, and then ask, were they capable of forming rosettes or were they defective in some way? And just to get a sense of, you know, how laborious this was, you know, she screened over 29, well, let's see, I'll do the quick math, 37,000 clones, whoops, and only got 18 mutants. But these mutants actually fell into some interesting classes. So here on the left, for comparison, is the wild type. So when it's not induced, it can be unicellular or it can form these linear chains of cells. And when it's induced, then it forms these beautiful rosettes. And what you can see is that there are a range of different phenotypes under the induced and the uninduced forms. This one was particularly interesting because it was perfectly normal in the uninduced state, but then entirely defective in rosette development. Many of the others were defective under both conditions, including this one, these clumpy cells in class D. They would clump up in the uninduced state and in the induced state. And when we, one of the neat things about rosettes is if you introduce shear, the rosettes are entirely robust to that. You can even vortex them and they stay together. Whereas these clumps, if you vortex them or even look at them the wrong way, they fall apart. So it's a much gentler interaction here than here. And so Tara Levin sought to characterize the genetics of this phenotype. And then later, another graduate student, Laura Wetzel, really focused on this class. Okay, the problem was that although we had mutants and we had interesting phenotypes, we didn't yet have a straightforward way to connect genotype to phenotype. And some of the key reasons were that we didn't know if choanoflagellates mated. And in fact, despite having been first identified in the mid 1800s, no one had seen choanoflagellate mating. And even if they had, they might not have known what they were looking at. So we didn't know if choanoflagellates could mate. We didn't know if that mating could be induced in the lab. And if they did mate, we weren't sure we could isolate the meiotic products. And without that, we didn't know if we could map the genetic lesions. We really needed the mating to essentially separate out the causative mutations from the background mutations. And keep in mind, I think that's the best way to do it anyway, but we didn't have CRISPR or any genome editing. So any other approach you might think about, we didn't have at our disposal. So really mating was a key barrier. So Tara decided to take that on also. And what's clear is that in many microbial eukaryotes, stressful conditions can induce mating. And so she started applying different stressful conditions to choanoflagellates. And sure enough, starvation, it turned out, was sufficient to induce mating. But even when the cells were starved, mating was rare. And this helped to explain why it hadn't been observed before. So in this whole field of cells, you can see that they're starved. They look, they're pointy and they look unhappy relative to the other cells you've seen so far. But of these, only 2 are about to mate, these 2 right here. And generally, we found that the cells differentiate in terms of size with a small cell and a large cell that we've provisionally named, the male and the female gamete. And so if we zoom in on those 2 cells, we can see how mating occurs. So the cells do this little dance. The male cell binds toward the apical pole of the cell, and then the cells fuse. And so this process has led us to, identify what we're calling the hallmarks of mating in choanoflagellates. And in this, what happens is the cells find each other. The membranes then fuse. After that, you get nuclear fusion, recombination, and then to return to the haploid state, there has to be a round of meiosis. And Tara identified all of those steps in choanoflagellates, including evidence for recombination. So that one of the key things to keep in mind at this point now is that these choanoflagellates have dynamic life histories. So I've told you that a single cell can become multicellular in terms of the rosette. And when the rosette inducing factors are not present, it can also form these chains. But this is all asexual. And these choanoflagellates can also land to produce these thecate cells, or during starvation, they form these small, what we call fast swimmers. But Tara uncovered another part of the life history, which is sexual reproduction, in which the haploid cells can fuse and then produce diploids that are thecate, and then they can return either into the asexual part of the life history or continue through a sexual reproduction pathway. So the discovery of mating then made it possible to map these rosetless mutants. And so the first thing that happened is Tara was able to map one of these class A mutants to a particular protein called the C-type lectin. And you can see it in teal here. It produced, this is the extracellular matrix that I mentioned before. And if this C-type lectin is not present, the choanoflagellate will not become multicellular, or it won't form these rosettes. So that really pointed to an important role for the extracellular matrix. And interestingly enough, when Laura focused on this class, she uncovered two different genes, two different glycosyl transferases that were also critical for the wild type rosette development as a choanoflagellate. And these also are genes that are involved in sculpting the extracellular matrix. So these two findings were very exciting. They, first of all, were a proof of principle that we could do forward genetics, but also pointed to an important role for the extracellular matrix that we really hadn't been focusing on before. However, neither of these two genes was the receptor for the rosette-inducing factor. And so I want to return to that hunt, okay? And here's where now Ariel Woznicka, a graduate student at the time, stepped in. So we couldn't use chemical approaches to tag the rosette-inducing factor. There were a number of reasons why that wasn't working. Forward genetics was not rapidly leading us to the receptor, although it's certainly an approach that's worth continuing to take. So, you know, in an act of desperation, in a way, we thought, what if we can use a comparative transcriptomic approach to identify genes that are specifically upregulated in rosette development cultures and not upregulated when they're not forming rosettes. And because because the inducing bacterium Algoraphagus is delivering lots of different cues to the choanoflagellates, not just the rosette-inducing factors, we thought we could sort of eliminate those background ones by also comparing S. rosetta cultures to cultures treated with other bacteria that induce rosettes. So here what you're seeing is a phylogeny of different bacteria, and then those that we know induce rosette development are shown with an X. So the idea was, are there specific genes that are upregulated in all of these cultures in the choanoflagellate and not upregulated when they're exposed to bacteria that don't induce rosette development? So I hope that's clear. So Ariel was and is an extremely thorough and thoughtful biologist. And so when she did these experiments, every time she treated with a different bacterium, she went back, she didn't accept, you know, that what she had been told that they don't induce rosette development. She went back and looked at them herself carefully. And when she did that, she saw something really weird in cultures treated with Vibrio fisheri. So sure enough, you know, we looked at Vibrio fisheri a lot. It's a well-known commensal and symbiont with different animals. We thought it would be super cool if it induced rosette development, but it didn't. But when she looked right away, so she would add the bacteria, and then rather than waiting 24 hours for rosette development to occur, she looked right away. And this is what she saw. So on the left, you see a culture that's been treated with a control bacterium that doesn't induce rosette development or anything, but fisheri, when added, caused the choanoflagellates to aggregate into these clumps. And these clumps are very different from rosettes because they're forming through different cells coming together. So those of you who know dictyostelium, you know, it's not as beautiful as dictyostelium, but it's conceptually similar with these cells coming together and aggregating. And so I still remember sitting in my office and Ariel coming to the door, and she's like, there's something weird going on in these cultures. I don't know what it is. You know, can you come look? And I looked, and we spent a bit of time trying to figure out what might be going on, because this wasn't a behavior that we'd seen in the lab before. Again, it had not been reported for choanoflagellates, and we didn't really have a strong notion of whether this was even physiologically relevant. That was like, Ariel was like, I don't know whether to keep studying it because I don't know if this is just some weird lab thing or if this is something that is actually indicative of S. rosetta's biology and nature. But then I remembered that many ciliates actually swarm before they mate. And in addition, swarming is actually a prominent behavior of mating in animals. And so you can see, fish swarm. These are these African lake flies that form these huge swarms, bats swarm, crab swarm. This is a common behavior where members of a species increase their local density prior to mating. And so we wondered whether that might be what's going on with S. rosetta also. And in fact, that was the answer. And so it turns out that Vibrio fischeri is not only inducing swarming and clumping, but it's also that is the lead-in to mating. And so here you can see two cells with their nuclei labeled. Their membranes fuse, their nuclei migrate together and fuse. And then remember, a key question is, do they undergo recombination? And so she tested that as well. And so here you see two strains with different genetic variants. And if she compared the outcome or the genotypes of bacteria that came from this, mixing these two together in the flask, whether they were exposed to control bacteria or Vibrio fischeri. And sure enough, with control bacteria, she only recovered the parental genotypes. But when she added Vibrio fischeri now, she saw abundant evidence for recombination. And so that told us that Vibrio fischeri is producing a molecule that induces mating in the choanoflagellate. And so just to summarize that here, this is a nice figure that accompanied a News and Views piece. And so the idea then is that when haploid S. rosetta is exposed to Vibrio fischeri is releasing a molecule which we've named Eros. And this is actually a chondroitinase, and that chondroitinase interacts with S. rosetta, triggering swarming and cells actually meeting up together. Those cells then fuse, they become diploid, and then through meiosis and recombination, return to a haploid state. So Ariel did not answer the question of what is the receptor for the resin-inducing factor, but she found this really remarkable life history activity. And this mating was actually in many ways different from the mating induced by starvation, because it happened almost right away, within minutes instead of within weeks. And the orientation of the cells was different, with the cells contact each other by the cell bodies, as opposed to fusing at the collar of the larger cell. And so this is an interesting phenomenon where we essentially have these two different mating pathways. Okay, so that used to be what I primarily talked about, but I want to return to this hunt for the receptor, because this is going to bring us to the new data that I'm very excited about. So this is the work of a postdoc in the lab, Alan Garcia de los Llanos. And here he is as we're trying to brainstorm and understand some new data he has, that some of which bears on what I've just been telling you. So when you look at the genome of the choanoflagellate, if you had to guess what the receptor for the rosette-inducing factor might be, the best possible candidate are these G protein coupled receptors. So why is that our best candidate? Well, in animals, G protein coupled receptors, first of all, they're the largest family of cell receptors, but importantly, although they can respond to many different kinds of inputs, they are the best known and best characterized receptors for lipids in general, but in particular, a special class of lipids called the sphingolipids. And if you remember, the rosette-inducing factor shares many chemical features in common with sphingolipids. And so we thought the G protein coupled receptors would be a good candidate. Now, before choanoflagellate genomes and transcriptomes were sequenced, it was thought that GPCRs were restricted to animals. But in fact, we now know that GPCRs have a much richer phylogenetic history. So this is just a schematic of one particular class of G protein coupled receptors. This is the adhesion G protein coupled receptors, which have adhesion motifs in this extracellular domain, a cleavage domain called a GPS, and then the transmembrane domain and intracellular motifs. And it turns out that GPCRs in general and adhesion GPCRs in particular are widespread across choanoflagellate diversity. And so here's our friend Salpingolica rosetta right here. And you can see that it has 19 total GPCRs and 12 of these adhesion GPCRs. And so this was very interesting. In addition, if we look across eukaryotic diversity, we see that animals have lots of adhesion GPCRs, as do choanoflagellates and other unicellular close relatives of animals. But the prevalence of GPCRs drops off dramatically as you get further away on the phylogenetic tree. So all of this led us to be very interested in not only in GPCRs in general, but adhesion GPCRs in particular. And what I'm going to do is focus on one particular class of GPCRs that we find is conserved across at least 9 different choanoflagellate species. So this GPCR has EGF domains near the N-terminus. It has a LAMG domain, and importantly, it has this GPS domain that's conserved. And for reasons that should become clear, we've named this GPCR in S. rosetta Clumpy. So here again is the full-length GPCR, and Ilan was able to generate edits at 2 different places. One right here, so this one will be called 1 through 553, and one right here, which is 1 through 95. And the key thing I want to tell you is that these 2 edits have the same phenotype in every case, and so we think these are knockouts. Okay, so what you're going to see is the cellular behavior of wild type cells here, and then 2 different mutants. And what I hope you'll be able to see is that these cells actually aggregate together over time. So here are our single cells, and now you can see these big clumps starting to form. Okay, perhaps reminiscent of something that you've seen recently. But we were surprised because Clumpy is an adhesion GPCR, so we expected lots of adhesion, not gain of adhesion. This is just to say that Ilan quantified this, and again, as you move through time with the wild type on the top and then the 2 mutants, you see that you accumulate these large clumps, and then you can quantify them, and you can see the wild type in black does not clump up, but these 2 mutants do, and essentially at equivalent rates. Okay, so that was unexpected. Is Clumpy important for growth? And we can find no difference in the growth curve of the control versus these 2 Clumpy mutants. Is Clumpy important for rosette development? It turns out it's not. So here's wild type on the top, no induction or with rosette induction. And you can see wild type forms beautiful rosettes, as do these Clumpy mutants. Okay, and it's quantified over here, showing that they are essentially equivalent. OMVs are outer membrane vesicles, and this is how we deliver the bacterial cues. So no difference. So far, the only difference that we can see is this aggregation behavior, but it doesn't seem to affect the cell adhesion that's required for rosette development. So how are these clumps forming? They're actually forming when cells are adhering through their collars. So these are not adhering to their cell bodies the way that Vibrio-induced mating cells were adhering. These are adhering through their collars. And Elan actually went through and measured the number of contacts of cells to collars, cells to cells, or collars to collars. And what he found is that the vast majority of the interactions were collar-collar or collar-cell. And in work I'm not showing you, when he removed the collars, he found that that completely blocked cell aggregation. Now, it turns out that this collar-collar interaction actually is similar to the type of interaction that we see in cells that are mating in response to starvation, not ones that mate in response to Vibrio treatment. And so here you can see pictures of the mutants or the control cells that have been starved. And when Elan did the same type of analysis of the number of contacts, when he looked at starved cells, this is a control edit here, he saw that they interacted in the same way. So primarily through collar-collar and collar cell body. So this certainly was very interesting and unexpected. And so he began to wonder whether in fact he had uncovered a negative regulator of mating. Could it be that by knocking out this GPCR, in fact, he was unleashing mating under conditions where mating didn't normally occur? And so to test that, he developed an assay for cell fusion that allowed him to detect rare fusion events using FACS. And so the idea was to use cells that were either labeled in the cytoplasm with MTFP or in the nucleus, a histone tag with mCherry. And then after 20 hours, measure the number of cells that had both fluorescent signals. And so here's one example here where you can see the cytoplasmic fluorescence, the nuclear fluorescence, and then the overlay. So this is in wild type cells. And the question was, if you did the same with these mutant cells, what would happen? And pardon me. In fact, there was a dramatic increase in the number of cells that were fusing. Here we're seeing the percentage of fused cells of the control versus Clumpy. And you can see that there is a dramatic and significant difference in the number of cells that are fusing in response to this Clumpy mutation. And so that suggested, in fact, that Clumpy might be related to the mating pathway. And so the next thing that Elan did was to look at which genes were upregulated and downregulated in response to deletion of Clumpy. And so here what you're seeing are the log fold changes of genes in control versus Clumpy. And what I hope you can see is that there are 28 genes over here that are significantly downregulated and 59 that are significantly upregulated. And when he looked at what they were, interestingly, the vast majority of those that were downregulated were retrotransposons, whose regulation is thought to be involved in gametogenesis in many animals. Now, in terms of upregulation, there was an important number of them involved in cyclic GMP signaling, and Elan has a whole nother story related to this, to his deep dive on these genes. But what I want to talk about right now is the fact that there were also genes that are known to be involved in cell adhesion and cell fusion in gametes. And one of those is a homolog of HAP2, which is an ancient gamete fusogen in eukaryotes. And so this is just to show all of the eukaryotes that express HAP2 and use it for gamete fusion. And so the way HAP2 works is it sits between the membranes of gametes, and it helps to set up the cell fusion that allows these cells to fuse. And you can see here, Chlamydomonas has HAP2 that's expressed near the apical pole of the cell, which is where cell fusion occurs during mating in Chlamydomonas. And also in Tetrahymena, we see enrichment of HAP2 in the area in which these two cells have fused. And so what Elan has shown is that this Clumpy mutant, well, let me back up and just say in wild type, when the wild type cells are grown in high nutrient conditions, they don't mate. They have very little collar adhesion, very little cell fusion, and very low expression of HAP2 and other regulators of gametogenesis. But when he deletes most of the Clumpy protein or gene, what he finds is now he gets rampant clumping of cells, which is a phenotype that we associate with mating. He gets increased collar adhesion, increased collar fusion or cell fusion, and increased gamete gene expression. And these phenotypes match with the mating phenotypes that occur when we starve S. rosetta. And what we don't know at this point is what happens to the Clumpy gene during starvation, but this is certainly something that he's investigating now. And so with that, I'm going to close and just say thank you to, incredible lab who kind of held it together during the pandemic, and we're joyfully emerging from the other side. And I'd be very happy to take questions that, bear on the many different threads that I threw out there to you. And I think that all bear on trying to understand how environmental cues coupled with intrinsic genetic circuits help us to understand how cell differentiation evolves. So thank you all.
Thank you. That was great.
Thank you.
And maybe let's see if trainees have questions first.
Great.
Raise your hand, virtually raise your hand or put a question in the chat. Looks like
Hi, thank you so much for a wonderful talk. I really enjoyed it. You just light up my whole day. I have two questions. The first question is, does the arrow change the expression of HAP2? I was just wondering the connection between the arrow that you found in your bacterial screening and with the HAP2 that you found in the cellular mechanism of mating. And then my second question is, in one of the videos you show, I think it's I-95, the clump. So basically the question I'm trying to get to is, I'm interested in the swamping behavior that you mentioned. It looks like some of the colonies has more movement than the others. Some of the colonies stay still, but some of the colonies seems to move around and then aggregate more. I was just curious about whether that means there's any fundamental differences between the colony, or do you think that would just show the random and then it's more population differences? Yeah, thank you so much.
Thank you. This is actually a great opportunity for me to introduce Alon, who I've invited to come along. So maybe he can wave his hands. I just had him turn on his camera. So in terms of your first question, is Eros turned upregulated in response, or sorry, is Clumpy upregulated following treatment with Eros? I actually don't think we know, but that's why I asked Alon to come along. If he has, do you know if have you looked?
No, we don't know. And I think, yeah, you were asking for HAP2 as well. And we have no idea. But that's definitely something that will be interesting to try to know for sure.
Yeah. Great question. And then the other question, I think you were referring to wild type cultures that have rosettes that behave in different ways. And we actually see a lot of difference in behavior depending on how many cells are in the rosette. So the large rosettes tend, if you think about it, there are all these competing forces with these flagella, but the rosettes are not perfectly spherical or perfectly symmetrical. And so what you can see is that depending on how, how far they are off of symmetry, you'll see different behaviors in response to. And another thing that happens is sometimes the colonies actually collect lots of bacteria on the collar. They're really good at prey capture, and that will make the ways in which they swirl and move through the water even more chaotic. So I don't know that we know, like, is that an adaptive thing? Has there been selection on it, or is it just a secondary response to biophysical features of the rosette? We don't know. Let's see, Dominica, is that Are you next?
Perfect. Hi, that was phenomenal talk. We were talking about it while you were talking.
Oh, good. Thank you.
So it's really interesting to see that multicellularity is not an obligate part of development, that you can be induced in culture. And I'm wondering, it's my understanding that the bacteria are things that choanoflagellates prey on.
Right.
And I've heard that being used as an argument for metabolism as an inductor of morphogenesis, right? So unicellular to multicellularity.
Right.
I just wonder if they're preying on bacteria and they're in taking them in, do they need receptors, or is it possible to do like conditioned media with what things bacteria produce? I don't know how that would work in culture.
Yeah. So we thought it was going to be a metabolic story, but it's not actually, because we can do conditioned media, but we can also just take that lipid at very, very low concentrations, and that alone is sufficient to induce rosette development. It doesn't seem to change the metabolism because they're eating a different bacterium. So they have plenty of nutrients, and all they need is this one little cue. And when we look at the concentration that is required, it seems to be far more consistent with a model of there being a receptor than with a metabolic model. In addition, there's another small molecule that we've isolated, which we've named inhibitor of rosettes. And like, what was it? IOR. And it essentially is a hydrolyzed version of RIF1, and it looks like it's acting as a competitive inhibitor. So that also, I think, is consistent with there being some sort of receptor. But maybe there's no receptor, and we're just hopefully going to keep learning cool things along the way. I don't know.
Interesting. Thank you.
Bluma, I'll let you go ahead and tell me who to.
I think the young man had his hand up, and then Art.
Okay, good.
It's very inspiring talk, and it make me, the light up the whole days about the mammalian development. So I was so curious about just why it's the specific lipid makes this kind of multicellular development. It seemed like this lipid is how to say, how to amplify the signaling, make them together, right? So, what's this receptor that in GPCR receptor or some other receptor of this specific lipid make this kind of signaling, make this cell aggregate, or we can say the development?
Yeah. I'm not sure I entirely understood the question, but I think I have a few things that might contribute to understanding. One is that it turns out that RIF1, that particular molecule, it's a highly specific interaction. And if we change the stereochemistry of that molecule at all, it doesn't work. So there's some sort of very specific interaction, but it's actually a very poor inducer of rosette development. And so it does induce, but at very low levels. And so Ariel Woznicka, the grad student I mentioned, actually went back and discovered that there are additional lipids, and she identified two others. One was similar to RIF1, and we called it RIF2 very creatively. And the second is a lysophosphatidylethynolamine that has no inducing activity on its own, but when mixed with the RIFs, they synergize, and it essentially recapitulates full induction, those three lipids together. And we don't understand that at all. So that's one source of specificity that occurs and boosting of signal. The other one that's very interesting is work by a former graduate student, Ella Ireland, in which she found that if you actually mix the mating inducing signal, Eros, with the rosette inducing ones, that greatly amplifies rosette induction also. And that we still don't understand. And I'd love to hear people's ideas of why that might be the case. But it seems that essentially she adds them both together, they mate really fast, and then they form these really big, beautiful rosettes, even at very low concentrations of the rosette-inducing factor.
But for example, so whether you can inhibit use the drugs like to inhibit a specific signaling to see whether it's blocked this kind of even you add the lipid there, you block this kind of development.
We've done a lot of inhibitor studies, and so far have not found an inhibitor that is specific to the process of rosette induction. But it's a good idea. You know what? I take that back. Now that I'm thinking about it, a former postdoc in the lab, Flora Rudigenera, did a really beautiful chemical genetic screen. And the first phase of that is now on bioRxiv, in which she was looking for inhibitors of cell proliferation. But she has a second wave of screen in which she has identified specific inhibitors of rosette development, and she hasn't yet fully worked that up. So I don't actually know which inhibitors they were, but she's in the process of working. She has her own lab now at Stanford, and she's working up those results now. So that's a very good point.
Okay, thanks. And another question, I'm sorry about two questions. And for this sexual, sexual reproduction, asexual reproduction there, is the rosette formation the similar or not? And also the for the sexual, they're mating together, and whether they form the synaptonema complex for the recombination. Okay.
We do not have enough resolution. These are very small cells, and we can't visualize even individual chromosomes at this point. We think they have over 50 chromosomes, and we just have not looked at that level of resolution, so we don't know. And the first question was about whether aggregation and colony development are the same thing, or?
Same. Sexuals and sexual, the rosette development is, the same, is similar or not?
No, they're completely different. So rosette development is clonal, and it's through cell division, and aggregation is cells coming together that are not related to each other necessarily.
What my question is, the sexual, because if you starvation the sexual, they mate with each other, and then they will move forward to this kind of rosette structure. But if you inducing the rosette asexual reproduction, they will also form the rosette structure. Both of this kind of rosette structure is similar or not, or they share the similar feature.
Rosette development is independent from mating.
That's
so they are not.
Okay, got it. Okay, thank you so much.
Right, sure. I think Art was next.
So biochemists often go to the brute force sort of avenue, and it seems to me that you could make RIF synthetically and make it radiochemically hot, and it'll be still functional. I'm sure of that. And just use that to just chase it down through chromatography.
I love that idea.
Dirty experiment that would be time consuming, but you'll definitely get, I would assume you can get conditions where hopefully RIF isn't itself metabolized, but is stably bound to its receptor.
I love it. Unfortunately, I can't get any of our collaborators to isolate that for us. So it's actually an extremely inefficient purification, and the synthesis. I'm trying to remember. The synthesis is extremely inefficient also. So unfortunately, that has not been an avenue that we've been able to go. We've also, our collaborators have been trying to tag the compound through synthetic approach.
No, I'm sure
that. That's not active.
No.
But the inhibitor is. So a tagged inhibitor is active, and so that is one possible route that we'll take in the future. But Yeah, it's a new generation. I've spent my time in the hot room.
How many steps of synthesis is it?
I'm afraid I don't remember.
Okay, sorry.
Since I didn't actually do it. So yeah, thank you though.
Okay, Lynn.
Hi, Nicole. Thanks for a great talk.
Thanks, Lynn.
I have a question about, you said you didn't have resolution to see chromosomes, and I'm wondering about cell divisions, because I'm curious about the difference between linear chains and rosettes. And is there, for example, are the linear chains also connected with bridges? Can you vortex them and they'll stick together? And then does something happen about the cleavage planes when the rosette is induced so that they form the more three-dimensional thing rather than the chain?
Right.
So can you watch that happening, I guess is the question.
So the linear chains are connected by bridges, but it is not a robust connection. So they will fall apart if vortexed. And we don't really, I think there's more work to be done, but this is my view of what's happening during rosette development. I think that when they divide, they remain, you know, it's symmetrical and you get two, but what they're doing is they've secreted this shared extracellular matrix. And I think that extracellular matrix tightens up. And that pulls the basal poles together and tilts the apical poles apart. And that view comes from a lot of very indirect research. And I think, you know, what we haven't done is a really careful examination of what happens when we treat with enzymes that might cleave the extracellular matrix. Will that, you know, prevent that? One of the challenges we have is that it's actually really difficult to keep the colonies in place under the microscope to watch the full process of rosette development. But yeah, these are open questions that I think we really need to get back and delve into more deeply.
Great, thanks.
Thanks.
I have one more question.
Sure.
Yeah, but I was wondering what happens if you mix Clumpy knockout with wild type. Do they just fuse with each other, or will they fuse with a?
Yeah, I took that slide out. Alon did do that experiment, and they actually will fuse. Or I don't think he's tested yet if they fuse, but they will adhere to the wild type cells. So it seems like it doesn't have to be bidirectional. It's enough to have a sticky mutant, and it will stick to the wild type cells.
Okay, any other questions? If not, let's thank Nicole again.
And thank you all for the great questions. That was fun.
And we'll, I guess at this point, log out of here, and there's a separate, you have a little break.
Okay.
Separate link for the meetings later.
Outstanding. Okay, I look forward to talking to all of you. Take care.
If you want, I know you were scheduled to have a 15-minute break, and it's now a 5-minute break. So, and I'm the next meeting. So if you want a little more time, we can.
Oh no, it's fine. I'm just gonna go and make a little more coffee, and then I'll meet you on the other one.
Sounds good.
Okay, take care.